Dear students, in the last video, as I have mentioned, we'll start with the biological classification. Biological classification, ಜೀವಿಗಳ ವರ್ಗೀಕರಣ. So in what way biological classification has been done over here? So the whole concept we'll study today. Topic is biological classification. And the meaning, bio means biological entities. Biological entities. These are nothing but organisms. So biological entities are classified over here. Classified over here. Classification andre ವರ್ಗೀಕರಣ. So ಈ ಜೀವಿಗಳನ್ನ ವರ್ಗೀಕರಣ ಮಾಡಿ ಯಾವ ರೀತಿಯಾಗಿ ಜೀವಿಗಳನ್ನ ವರ್ಗೀಕರಣ ಮಾಡ್ತಾರೆ ಈ ಚಾಪ್ಟರ್ ಅಲ್ಲಿ ಕಂಪ್ಲೀಟ್ ಆಗಿ ನೋಡ್ತಾ ಹೋಗೋಣ. ರೈಟ್, ಟೋಟಲಿ ಆಸ್ ವಿ ನೋ 5 to 30 million varieties of millions of species are existing on this earth. It is being expected by the scientists. 5 ರಿಂದ 30 ಮಿಲಿಯನ್ ಸ್ಪೀಸೀಸ್ ಪ್ರಭೇದಗಳನ್ನ ಈಗಾಗಲೇ ನಾವು ಎಕ್ಸ್ಪೆಕ್ಟೇಷನ್ ಮಾಡ್ತೀವಿ ಅಷ್ಟೊಂದು ಪ್ರಭೇದಗಳ ಭೂಮಿ ಮೇಲೆ ಬದುಕಿದಾವೆ. ಔಟ್ ಆಫ್ ದೀಸ್ 1.7 has been identified. Out of them 1.7 millions of organisms have been identified. So among them, both animals as well as plants are present over here. Right? 1.7 millions. It includes both animals as well as plants. Right? So this many number of organisms, what we have already identified, classified, even named, ಅವಕ್ಕೊಂದೊಂದು ಹೆಸರುಗಳನ್ನು ಕೂಡ ಕೊಡಲಾಗಿದೆ. ಇಷ್ಟು ಜೀವಿಗಳನ್ನು ಕಂಡುಕೊಂಡಿದ್ದೀವಿ. ಇನ್ನು ನೋಡಿದ ಜೀವಿಗಳು ಅದಾವ ಅಂತ ಹೇಳಿ ಎಕ್ಸ್ಪೆಕ್ಟೇಷನ್ ಮಾಡಿದ್ದಾರೆ, ಆದರೆ ಅವು ಇನ್ನೂ ಐಡೆಂಟಿಫಿಕೇಶನ್ ಮಾಡಬೇಕಾಗಿದೆ. ರೈಟ್? ಸೊ ಹೌ ಟು ಸ್ಟಡಿ ದಿಸ್ ಮೆನಿ ನಂಬರ್ ಆಫ್ millions of organisms? The question arises. It is very difficult to study individual organisms. ಪ್ರತಿ ಜೀವಿಗಳನ್ನು ಅವುಗಳ ಗುಣಲಕ್ಷಣಗಳನ್ನ ಅವುಗಳ ಸೈಜು ಇವುಗಳನ್ನೆಲ್ಲ ಅಭ್ಯಾಸ ಮಾಡೋದು ನೆನಪಿಟ್ಕೊಳ್ಳೋದು ಕಷ್ಟ ಆಗ್ತದೆ. ಫಾರ್ ದಟ್ ರೀಸನ್ ದ ಮೆಥಡ್ ಆಫ್ ಕ್ಲಾಸಿಫಿಕೇಶನ್ ಕಮ್ ಟು ಎಕ್ಸಿಸ್ಟೆನ್ಸ್. ರೈಟ್? ಅಂದ್ರೆ ವರ್ಗೀಕರಣ, ಜೀವಿಗಳನ್ನು ವರ್ಗೀಕರಣ ಮಾಡಬಹುದು. So the animals or the plants which have common characteristic features, morphological characters, internal anatomical characters, ಈ ಒಂದು ಗುಣಲಕ್ಷಣಗಳನ್ನ ಒಟ್ಟಾಗಿ ಕನ್ಸಿಡರೇಷನ್ ಮಾಡಿ ಯಾವ ಪ್ರಭೇದದ ಜೀವಿಗಳಲ್ಲಿ ಕಾಮನ್ ಆಗಿ ಬರ್ತವ ಅವುಗಳನ್ನ ಒಂದು ಗುಂಪಾಗಿ ನಾವು ಮಾಡ್ತೀವಿ. ದಟ್ ಈಸ್ ದ ವೇ ಹೌ ದ ಬಯೋಲಾಜಿಕಲ್ ಕ್ಲಾಸಿಫಿಕೇಶನ್ ಹ್ಯಾಸ್ ಬೀನ್ ಡನ್. ಅಂಡ್ ಇಟ್ ವಾಸ್ ಇನಿಷಿಯೇಟೆಡ್ ಬೈ ದ ಫಸ್ಟ್ ಸೈಂಟಿಸ್ಟ್ Aristotle. Aristotle is the earliest person, the one who had started the classification of organisms. He considered their most common morphological characteristic features, easily identifiable. So characteristic features have been considered by him for classification of organisms. ಜೀವಿಗಳನ್ನು ವರ್ಗೀಕರಣ ಮಾಡಲಿಕ್ಕೆ ಬಹಳಷ್ಟು ಹತ್ತಿರದ ಅಥವಾ ಬಹಳ ಸಂಬಂಧಪಟ್ಟಂತಹ ಗುಣಲಕ್ಷಣಗಳನ್ನು ಕನ್ಸಿಡರೇಷನ್ ಮಾಡಿಕೊಂಡು ನಾವು ವರ್ಗೀಕರಣ ಮಾಡಿ. ಎಕ್ಸಾಂಪಲ್, animals have been classified into two groups by Aristotle. Aristotle classified animals into two groups. One is Anima. Are the two groups of animals he identified. So these are the animals with red blood. These are the animals with the red blood. These are the animals without the red blood. So this way he classified animals into two groups. So whether the animals have red blood or not, based on that he classified the animals. In the same way he tried to classify the plants by external features, whether the plants are herbs, shrubs, as well as trees. This way, he classified the plants, herbs, shrubs, as well as trees. This way, he tried to classify the organisms. So, by most common identifiable characteristic features, he considered, right? So, next one. Yes, the father of classification. Father of classification. You might the name of this scientist, Carolus Linnaeus. Carolus Linnaeus is the name of the scientist, the one who is called as father of classification. Why? Because he is the first person, the one who initiated. So, by considering the most common characteristic features, and he tried to classify maximum number of species, and he identified maximum number of species. For that reason, he is called as father of classification. He classified all the organisms on this earth into two major groups, right? One is animals and the second one is plants. And these were kept in one kingdom. As you know, kingdoms, under samrajyagalu, so animals are all kept in one kingdom, one samrajyadalli. All the plants were kept in one kingdom. So hence they are given the names as kingdom Animalia and kingdom Plantae are the two kingdoms he identified, right? So these were kingdoms, right? ಎರಡು ಸಾಮ್ರಾಜ್ಯಗಳನ್ನು. Hence, this system of classification is called as two kingdom classification. Two kingdom classification was given by Carolus Linnaeus. He made two kingdoms. So, by classifying all the organisms on this earth as Animalia as well as Plantae, two kingdom system was introduced by Carolus Linnaeus. So, next one, Ernst Haeckel. Ernst Haeckel is another scientist, the one who introduced a new kingdom, right? So that is Protista. Protista was introduced by Ernst Haeckel. Hence he was called as the scientist, the one who introduced the three kingdom classification. After himself, another scientist, Copeland, is the name of the scientist, the one who introduced one more kingdom, kingdom Monera. These are all primitive organisms he introduced, and it became a four kingdom classification. Four kingdom classification. And totally, R.H. Whittaker is the name of the scientist, the one who made five kingdom classification. R.H. Whittaker is the name of the scientist, the one who made five kingdom classification. Ultimately, all those kingdoms, when we gather, that moment, first one is Monera, and the second one is Protista, third one is Mycota, fourth one is Metaphyta, and fifth one is Metazoa. Like this, five kingdom classification has been done ultimately, So first kingdom is kingdom Monera, kingdom Protista, kingdom Mycota, kingdom Metaphyta is called as kingdom Plantae, and Metazoa is called as kingdom Animalia. So this way, so totally there are 5 kingdoms were made ultimately by R.H. Whittaker, right? So these 5 kingdoms so are not exactly try to classify all the organisms into several groups or 5 groups over here. Many of the merits and demerits are there in 5 kingdom classification also. The best example for this, I would like to mention one example over here. One example is viruses. ಇವೆಲ್ಲ ಗ್ರೂಪ್ಗಳನ್ನು ನೀವು ಕನ್ಸಿಡರೇಷನ್ ಮಾಡ್ರಿ. ಇವು ಯಾವುದಾದ್ರೂ ಗ್ರೂಪ್ ಅಲ್ಲಿ ವೈರಸಸ್ ಬರ್ತಾವ ನೋಡ್ರಿ. There are no viruses included in any of the groups. Then this classification method is not complete one. It has many drawbacks. It has many merits even. Right? In the same way, Euglena, mixotrophic animal ಅಂತ ಹೇಳಿ ಕರೀತೀವಿ ನಾವು. So why? Because it has chlorophyll pigment. It can synthesize its own food material sometimes. Sometimes it acts as heterotrophic animal, means it feeds on other animals. Like this. So this animal is included in Protista since it is unicellular organism. Unicellular eukaryotic organism ಅಂತ ಹೇಳಿಬಿಟ್ಟು ಇದನ್ನ ಪ್ರೊಟಿಸ್ಟಾ ಒಳಗಡೆ ಸೇರಿಸಿದ್ರು. ಆದರೆ ಆಕ್ಚುಲಿ ದಿಸ್ ಈಸ್ ಅನ್ ಅನಿಮಲ್. ಅದು ಒಂದು ಅನಿಮಲ್ ಆಗಿದೆ. ಸೊ ಇಟ್ ಹ್ಯಾಸ್ ಟು ಬಿ ಅಡೆಡ್ ಇನ್ ಅನಿಮೇಲಿಯಾ. ಬಟ್ ಅನಿಮೇಲಿಯಾದಲ್ಲಿ ಸೇರಿಸಲಿಲ್ಲ, ಅದರ ಬದಲಾಗಿ ಪ್ರೊಟಿಸ್ಟಾದಲ್ಲಿ ಸೇರಿಸಲಾಯಿತು. Such kind of many drawbacks are there in 5 kingdom classification. So, but still, so this is most acceptable method of classification. Let us start with one by one the characteristic feature of all the kingdoms as we have studied in the previous classes. The first kingdom is kingdom Monera. As earlier I have mentioned, these Monerans are the most primitive organisms. They are microscopic in nature, right? So the general characteristic feature of these Monerans, as I say, these are also said to be as prokaryote. I hope ನಿಮ್ಗೆ ಗೊತ್ತಾಗಿರಬಹುದು. prokaryote ಅಂತ ಬರೆದಾಗ Monerans ಅನ್ನ one characteristic feature is very clear to you. What is that one characteristic feature? All these organisms are primitive organisms. They do not have any particular nuclear membrane around the nucleus, and that is the peculiar characteristic feature you can understand over here. So then we'll write the characteristic feature one by one. These are microscopic unicellular, microscopic unicellular prokaryotic organisms, and these are represented by naked DNA, right? Naked DNA means, already I have mentioned, this is called as nucleoid or incipient nucleus. Incipient nucleus, or it is also known as nucleoid. Nucleoid it is also called as. This is the peculiar characteristic feature of these Monerans, where absence of nuclear membrane is particularly seen over here. Here, absence of nuclear membrane is a peculiar characteristic feature of all these Monerans. And where these organisms are available. These are cosmopolitan in nature. What do you mean by cosmopolitan? Cosmopolitan means these organisms are available everywhere, in the sense that, so these organisms may be seen in deserts, they may be seen in colder regions, deep ocean floor. These are seen in deep ocean floor even, right, at the bottom of the sea. These are available. And these are also available in hot springs. In hot springs, they can be available, right? And these are available, or these are living in extreme conditions like high temperature. In the region where high temperature, it is about more than 80 degrees centigrade, they can live in, right? And they can exist in high salt condition. It is also mentioned to be as a salinity. In high salinity condition, they can survive themselves, right? In such extreme conditions, even in absence of oxygen, they can survive. Anaerobic condition, they can survive it. In these extreme conditions, all these monerans can exist themselves. Best examples for monerans, prokaryota. If I mentioned, I have already given examples. They are nothing but bacteria, right? Bacteria I have already mentioned in the previous class. Okay. So these are cosmopolitan in nature. Next one. In the previous class, as I had mentioned, these monerans are bacteria as cell wall, and that cell wall is made up of peptidoglycan. Cell wall is made up of peptidoglycan. Cell wall. And some of these have flagella. Flagella is the locomotory organs in these organisms. Flagella is made up of two globular proteins. Flagella are made up of two globular proteins, and these proteins are called as flagellin. Proteins are known as flagellin, right? And apart from this one, their nutrition, right? Nutrition. How do they prepare their own food material? Or so else they are acting as heterotrophs. See here, some are photosynthetic in nature. Some are photosynthetic in nature, some are chemosynthetic in nature, and some others are symbiotic in nature. Some are photosynthetic in the sense that photosynthesis is performed by some of the organisms. They can prepare their own food material, right? Patra harit irtada, chlorophyll pigment irtade ivugalali, ಅದಕ್ಕಾಗಿ ತಮ್ಮ ಆಹಾರವನ್ನು ತಾವೇ ಸಂಸ್ಕರಣೆ ಮಾಡಿಕೊಳ್ಳುವ ಅಂತವು ಈ ಆರ್ಗಾನಿಸಮ್ಸ್ ಆಗಿರ್ತವೆ. Apart from this one, chemosynthetic, chemicals, by using chemical components they prepare their own food material. Chemosynthetic organisms, they are called as monerans, are chemosynthetic in nature. Symbiotic in nature. Symbiotic is one type of method of nutrition they get, mutually helping each other. Two organisms live together and mutually they help each other. ಇದಕ್ಕೆ ಬೆಸ್ಟ್ ಎಕ್ಸಾಂಪಲ್ ಹೇಳಬೇಕಾದ್ರೆ ಒಂದು ಬೆಸ್ಟ್ ಎಕ್ಸಾಂಪಲ್ ರೈಜೋಬಿಯಂ ಬ್ಯಾಕ್ಟೀರಿಯಾ. ರೈಜೋಬಿಯಂ ಬ್ಯಾಕ್ಟೀರಿಯಾ, ಈ ಬ್ಯಾಕ್ಟೀರಿಯಾ ಇದು ರೂಟ್ ನೊಡ್ಯೂಲ್ಸ್ ಒಳಗಡೆ ಇರತಕ್ಕಂಥದ್ದು. ಲೆಗ್ಯೂಮ್ ಪ್ಲಾಂಟ್ಸ್ ಅಂತ ಹೇಳಿ ಕರಿತೀವಿ, ಲೆಗ್ಯೂಮಿನಸ್ ಪ್ಲಾಂಟ್ಸ್ ಅಂತ ಹೇಳಿ ಬರ್ತವ. ಆ ಲೆಗ್ಯೂಮ್ ಪ್ಲಾಂಟ್ಸ್ ಗೆ ಬೆಸ್ಟ್ ಎಕ್ಸಾಂಪಲ್ ಪೀ ಪ್ಲಾಂಟ್ ಅಂತ ಹೇಳ್ಬೋದು. ಇಂಥ ಪ್ಲಾಂಟ್ಸ್ ಅಲ್ಲಿ ರೂಟ್ ನೊಡ್ಯೂಲ್ಸ್ ಒಳಗಡೆ, ರೂಟ್ಸ್ ಒಳಗಡೆ ಗಂಟುಗಳಿರ್ತವ, ಅದ್ರ ಒಳಗಡೆ ಈ ಬ್ಯಾಕ್ಟೀರಿಯಾ ಕಂಡುಬರ್ತದೆ. ಇದು ನೈಟ್ರೋಜನ್ ಅನ್ನ, ಅಟ್ಮಾಸ್ಫಿಯರಿಕ್ ನೈಟ್ರೋಜನ್ ಅನ್ನ ಆ ಒಂದು ಸಸ್ಯಕ್ಕೆ ಪ್ರೊವೈಡ್ ಮಾಡಿದ್ರೆ ಆ ಸಸ್ಯದಿಂದ ಇದು ನ್ಯೂಟ್ರಿಷನ್ ಅನ್ನ, ಆಹಾರವನ್ನು ಪಡೀತದ ಮತ್ತು ಅಲ್ಲಿ ಆವಾಸ ಸ್ಥಾನವನ್ನು ಪಡೀತದೆ. ಲೈಕ್ ದಿಸ್ ಎರಡು ಆರ್ಗ್ಯಾನಿಸಮ್ಸ್ ಮ್ಯೂಚುವಲಿ ಏನು ಬೆನಿಫಿಟೆಡ್, ಎರಡು ಒಂದಕ್ಕೊಂದು ಸಹಾಯ ಮಾಡಿಕೊಂಡು ಕೂಡಿ ಇರ್ತವೆ. ಕೂಡು ಜೀವನ ಅಂತ ಕನ್ನಡದಲ್ಲಿ ಕರೀತಾರೆ. ದಟ್ ಕೈಂಡ್ ಆಫ್ ಲೈಫ್ ಇಸ್ ಲಿವಿಂಗ್ ದಟ್ ಇಸ್ ಸಿಂಬಯಾಟಿಕ್ ಲೈಫ್ ದೇ ಆರ್ ಲೀಡಿಂಗ್. ಅದಕ್ಕೂ ಕೂಡ ಆ ರೀತಿಯಾದಂತಹ ಒಂದು ನ್ಯೂಟ್ರಿಷನ್ ಅನ್ನ ಈ ಆರ್ಗ್ಯಾನಿಸಮ್ಸ್ ಹೊಂದಿರ್ತವೆ. ನೆಕ್ಸ್ಟ್ ಒನ್ nitrogen fixation. Nitrogen fixation is the unique feature of these monerans. Monera group ನಲ್ಲಿ ಬರ್ತಕ್ಕಂಥ ಬ್ಯಾಕ್ಟೀರಿಯಾಸ್ ಇರಬಹುದು, ಬ್ಲೂ ಗ್ರೀನ್ ಆಲ್ಗೇಸ್ ಇರಬಹುದು. ಸೊ ಇದಕ್ಕೆ ಎಕ್ಸಾಂಪಲ್ಸ್ ಅನ್ನು ನಾನು ಆಲ್ರೆಡಿ ಮೆನ್ಷನ್ ಮಾಡಿದ್ದೀನಿ. ಬ್ಲೂ ಗ್ರೀನ್ ಆಲ್ಗೇಸ್ ಬ್ಯಾಕ್ಟೀರಿಯಾಸ್, they have one peculiar characteristic feature. What is that peculiar characteristic feature? Is nitrogen fixation. ಆಲ್ರೆಡಿ ನಾನು ಒಂದು ಎಕ್ಸಾಂಪಲ್ ಅನ್ನು ಕೂಡ ಮೆನ್ಷನ್ ಮಾಡಿದ್ದೀನಿ. ರೈಜೋಬಿಯಂ ಬ್ಯಾಕ್ಟೀರಿಯಾ ಅಟ್ಮಾಸ್ಫಿಯರ್ ನಲ್ಲಿ ಇರತಕ್ಕಂಥ ನೈಟ್ರೋಜನ್ ಅನ್ನ ಸಾಯಿಲ್ ಅಲ್ಲಿ ಫಿಕ್ಸ್ ಮಾಡಿ ಆ ಪ್ಲಾಂಟ್ ಗೆ ಹೆಲ್ಪ್ ಮಾಡ್ತದೆ. ರೈಜೋಬಿಯಂ ಇಸ್ ನಾಟ್ ಓನ್ಲಿ ಎ ಸಿಂಗಲ್ ಎಕ್ಸಾಂಪಲ್, ರೈಟ್? ಬ್ಲೂ ಗ್ರೀನ್ ಆಲ್ಗೀಸ್ ಆರ್ ಆಲ್ಸೋ ದೇರ್. ನೀಲಿ ಹಸಿರು ಶೈವಲಗಳಂತ ಏನ್ ಕರಿತೀವಿ, ಇವು ಕೂಡ ಅಟ್ಮಾಸ್ಫಿಯರ್ ಅಲ್ಲಿ ಇರತಕ್ಕಂಥ ನೈಟ್ರೋಜನ್ ಅನ್ನು ಸಾಯಿಲ್ ಅಲ್ಲಿ ಫಿಕ್ಸ್ ಮಾಡ್ತಕ್ಕಂಥ ಒಂದು ಕೆಲಸವನ್ನು ಮಾಡ್ತವೆ. ಅದರಿಂದ ಸಸ್ಯಗಳಿಗೆ ಏನು ನೈಟ್ರೋಜನ್ ಸಿಗ್ತದೆ. ಎಲ್ಲಿ ನೈಟ್ರೋಜನ್ ದೊರಕೋದಲ್ಲ ಅಲ್ಲಿ ಈ ಆರ್ಗ್ಯಾನಿಸಮ್ಸ್ ಅವುಗಳಿಗೆ ನೈಟ್ರೋಜನ್ ಅನ್ನು ಕೊಡುವಂತ ವ್ಯವಸ್ಥೆಯನ್ನ ಮಾಡ್ತವೆ. ನೈಟ್ರೋಜನ್ ಫಿಕ್ಸೇಷನ್. ಸೊ ಬ್ಯಾಕ್ಟೀರಿಯಾಸ್ as well as blue green algae. So these are the best examples which are called as nitrogen fixers. Nitrogen fixers, these are called as, right? Last characteristic feature is reproduction. Reproduction, santanotpatti anta ನಾವು ಏನು ಕರಿತೀವಿ. These organisms, how do they reproduce their young ones? Reproduction. Very simplest form of reproduction they exhibit over here. Bhಳ simple ಆಗಿರತಕ್ಕಂಥದ್ದು. One is asexual type of reproduction. Asexual type of reproduction, as well as sexual type of reproduction. ಇವು ಎರಡು ಮೆಥಡ್ ನಿಂದ ಮೇನ್ಲಿ ಈ ಆರ್ಗ್ಯಾನಿಸಮ್ಸ್ ರೀಪ್ರೊಡಕ್ಷನ್ ಅನ್ನ ಮಾಡ್ತವೆ. One is asexual. As the name says here, it is a type of reproduction in which formation and fusion of gametes do not take place. ಈ organisms ಅಲ್ಲಿ ಈ type of reproduction ಅಲ್ಲಿ formation and fusion of gametes do not take place. ಹಾಗಾದ್ರೆ ಯಾವ ರೀತಿಯಾಗಿ ಇವು ಮಾಡ್ತವೆ? ಎಕ್ಸಾಂಪಲ್ ಅನ್ನು ನಾವು ತೆಗೆದುಕೊಂಡಾಗ ಬ್ಯಾಕ್ಟೀರಿಯಾಸ್ ಎಕ್ಸಾಂಪಲ್ ತಗೊಂಡಾಗ ಬೈನರಿ ಫಿಷನ್, ಇದು ಒನ್ ಮೆಥಡ್ ಆಫ್ asexual reproduction. ಬೈನರಿ ಫಿಷನ್ ಬಗ್ಗೆ ನೀವು ಈಗಾಗಲೇ ಓದಿದಿರಿ ಹಿಂದಿನ ಕ್ಲಾಸ್ ಅಲ್ಲಿ. ಒನ್ ಸೆಲ್ ಇದ್ದದ್ದು ಎಕ್ಸಾಕ್ಟ್ಲಿ 2 ಸೆಲ್ ಆಗಿ ಡಿವೈಡ್ ಆಗ್ತಕ್ಕಂಥದ್ದು ಮೈಟೋಟಿಕಲ್, ರೈಟ್? ಬೈನರಿ ಫಿಷನ್ is one method of reproduction. Yes, binary fission, you might know, single cell divides into two equal sized cells. Binary fission occurs usually in bacteria. In bacteria, it occurs over here. Apart from this one, they can form spores formation. spore formation is also another method of asexual type of reproduction in monerans, right? And sexual type of reproduction that occurs over here, which is a primitive method in a bacteria. Usually one is conjugation method of reproduction that we come across in bacteria, right? So here two bacterial cells, this is plus, this is minus. This kind of strained bacteria would be there. When they come very close to each other, that moment they form one conjugation tube between themselves, right? And this genetic material that are transferred to this one, then it becomes what? Positive. Like this method of conjugation method of reproduction that takes place, transduction as well as transformation. These are different methods of reproduction that we come across over here. In POC we study these one in detailed way, right? So you haven't studied in SSLC. ಇವುಗಳ ಬಗ್ಗೆ ಜಸ್ಟ್ ಒಂದು ಇಂಟ್ರೊಡಕ್ಟರಿ ಮಾಹಿತಿಯನ್ನು ಅಷ್ಟೇ ಕೊಟ್ಟಿದ್ದೀನಿ. ಹೆಸರುಗಳು ನಿಮ್ಗೆ ಗೊತ್ತಿದ್ರೆ ಸಾಕು, ಅದಕ್ಕಿಂತ ಹೆಚ್ಚಿಗೆ ಮಾಹಿತಿ ಬ್ರಿಡ್ಜ್ ಕೋರ್ಸ್ ಅಲ್ಲಿ ಅವಶ್ಯಕತೆ ಇಲ್ಲ ಅಂತ. ಥ್ಯಾಂಕ್ ಯು.